The overall goal of the following experiment is to observe the effects of different pulsatile flow pathologies on endothelial cells in vitro. This is achieved by first chemically treating glass slides and coating them with protein to ensure cell attachment during exposure to flow. As a second step, the pulsatility index of the flow is determined, which allows for comparison of in vitro flow to values described in the literature. Next, cell-seated slides are connected to the flow system in order to measure cell response to flow pathologies. The results show changes in morphology and protein expression based on flow profiles using optical microscopy and immunofluorescent staining.
The main advantage of this technique over the peristaltic or pulsatile pump is the ability to adjust flow pulsatility to mimic diseased or healthy response. This can help answer key questions in the field of arterial disease.
To begin, place new glass slides measuring 75 mm by 50 mm by 1 mm into a clean glass slide staining dish. Then, add enough of a 20% sulfuric acid solution to fully immerse the glass slides and incubate them in the acid solution overnight. The following day, wash the slides 3 times by immersing them in fresh deionized water for 5 minutes each time. Once rinsed, immerse the slides in acetone for 30 minutes, and then overnight in a solution of 6% 3-aminopropyl triethoxysilane in acetone. Next, rinse the slides 3 times in pure acetone for 5 minutes, changing acetone after each wash. Then, wash slides 3 times in deionized water for 5 minutes, changing the deionized water after each wash. Next, immerse slides